Suyash Sagar Bajpai, course coordinator of this 15 days research methodology add-on course, welcomes you all in the first session of day 11. I would like to invite Ms. Ragini Pyasiji to introduce our resource person of first session to our participants.
Hello, am I audible? Hello,
Yes,
am I
you.
audible? Hello?
Yeah, yes, you are audible.
Yes, yes, yes. Luxury and lies have huge maintenance costs, but truth and simplicity are self-maintained without any cost. Good afternoon, everyone. A very warm welcome to all of you. Thank you for having me. Myself, Ragini Pyasi. It's my privilege and honor to introduce our guest speaker today. He is a highly accomplished scholar and a researcher. Our speaker is holding an MSc and MPhil and PhD in statistics from Dr. BRA University with 23 years of experience. He has qualified NET UGC for lectureship and in present he is working as an assistant professor in JNU Delhi. Our speaker is a life member of International Indian Statistical Association, Indian Society of Medical Statistics, and Indian Society of Agricultural Statistics, and Data Safety Monitoring Board member for Phase 1 study of Pharmace Private Limited and for Phase 1 study of Panacea Biotech Private Limited. Exceeding all this, our speaker's best peer reviewed publications were Continuity in Fine Topological Space in International Journal of Science and Research, Evolutionary Analysis of Dengue Serotype 2 viruses using phylogenetic and Bayesian methods, Role of Herbs in Preventing Cancer, International Research Journal of Pharmacy, Microarray Data Analysis of Acute LLTR using statistical softwares. Eventually, it is my privilege to welcome a scholar and a researcher whose life's work has considerably contributed to statistics. Please join me in extending a warm welcome to Professor Manoj Kumar, sir. Thank you.
Sir, you are not audible.
सर आवाज नहीं आ रही।
Can you hear me now?
Yes, sir. Yes, sir.
Can you hear me?
Yes, sir.
Yes, sir.
Okay. So fair enough. I'm connecting with my mobile. Maybe some issue with my laptop. We will look later on. All right, so we was talking about the sample size estimation is a very important part of any research. Why it is important? Because everyone wants to interested that how much sample size is required to get the answer of their objective. Okay, so it's very important. You should needs to take out the number of samples appropriately. So we will talk about the how you can bring out about the sample size estimation. So we start with the research process. So what is a research process? So it's a journey of any research which is start with the what is your research problem. So basically what you would like to work, and then after that once you know that this is your research problem, and then after that you will talk about the concept and the theories, review concept and theories. Then you will looking the various articles, various reports, which is already published in your research problem area. Then after that you will formulate the hypothesis. So once you will formulate hypothesis based on your objective, then you will talk about the design research, which include the sample design or which include the sample size estimation. So here I would like to emphasize over this design research. Here you are interested that how to get the sample size, and then after that you will go for the collections and analysis of data. So basically whatever the objective is was with you, then you are interested to know that how you can get the answer of those objectives. Then you will talk about the interpret and the reports. Okay, so this is the research process which we are bringing out in any research work or in the thesis or in any projects. Okay, then we will talk about the basic specific problems. Okay, so here I'm trying to explain you. Let's say in any research you will talk about the some questions. Okay, so what questions you are interested to know, uh, or you are interested to get the answer of your questions. So let's say you will have one question or two question, maybe three questions in your research, and then you will talk about the experimental design. So what kind of the experimental design you will keeping in your research, and then you should know in the very beginning of the designing of your study, what kind of the statistical tests you will be using. Then after that you will talk about the sample size. So sample size, once you know your objective, and then you know the what kind of the design you will be using, and then you also knowing that statistical test is required, then you will talk about the sample size estimations. And then after that you will talk about the experiment, data collections, and data explorations, data analysis, and the results. So this is basically the cycle. And then after that the results you will get, and then after that you can able to give the answer of your questions, what happened to the your in this research cycle. So basically this is a research cycle where we will talk about over the all points, but here I would like to emphasize only on the sample size. So it's a you know that if you want to work on the particular genomic research, or let's say you would like to work on the particular therapeutic area, then it is there is a huge population is there. Okay, if you would like to know the what is the impact of the COVID-19 on our economy, then this is a larger populations. Okay, and you must know interested to know that how much small sample size or maybe appropriate sample size you require to get the answer of your research questions. So this term is coming up the sample size. So sample size is what are the good number which is required to get the answer of your research questions. Okay, so the sample size is basically depend on the sum of the factors. So let's say if it is,
सर सर
yeah.
प्लीज हिंदी में बता दीजिए, क्योंकि बहुत सारे बच्चे जो हैं वो हिंदी मीडियम ही हैं।
Okay, okay, all right. So it's sometimes okay. So sample size means how much number of the subject means, um, how much रेस्पोनेंट यू रिक्वायर्ड, आपको कितने लोगों से एक्सपेरिमेंट कराने की ज़रूरत है. इफ़ यू वुड लाइक टू नो if you will have any project in the laboratory, or maybe you want to go for survey, then how much number of the people will required in any study, that is the sample size. So the more heterogeneous population is larger samples needs to be. Okay, so if in the population जब आपका जो भी दायरा है उसमें अगर हेट्रोजीनियस लोग हैं, okay, heterogeneous means there is a heterogeneity of populations, means some are very high income people, some are very low income peoples, okay. So you will saying that it is a heterogeneous populations. So the larger the sample needs, if it is a heterogeneous, depend on the topic and frequently it occurs. The probability sampling, the larger sample size, the better would be your estimate. अगर आपका सैंपल साइज बड़ा होगा तो आपका जो भी आउटकम आएंगे, जो रिजल्ट्स आएंगे, दैट इज मोर अच्छे रहेंगे, दैट इज मोर एफिशिएंट और इफेक्टिव outcomes will come up. Okay, with the non-probability sample, not generalizable, but still the stability of the result is required. So what are the factors that influence the sample size calculations? आप कितना sample size calculate करेंगे उसके लिए there are the some factor which is affecting. So I think you gone through, I think I'm expecting that all of you are gone through the hypothesis testing, and then after that you might be knowing that desired level of significance. Okay, so desired level of significance, so these are the factors which is affecting the sample size calculations. जब आप करते हैं sample size को calculate, तो उसके लिए आपको ये सारी चीजें पता होना चाहिए और कैसे इसको impact करते हैं calculation में, ये हम देखना चाहेंगे इधर. So desired level of significance, desired power, and one tail or two tails. Okay, either it is a one tail or two tails, that is an important one. Then smallest differences or effect. Okay, so differences, you will be looking that is there any differences between the two samples or not. So smallest clinically important differences that investigated thinks is worth detecting, and differences that investigated think is likely to be detected. Okay, would an increase or decrease in the effect size make a significant clinical differences? Justifications of the previous data, how, what kind of the information how you will, what are the things is required. So published data, if it is any article is already published, जो वर्क है उसके रेफरेंसेस हम लेते हैं सैंपल साइज कैलकुलेट करने में, या प्रीवियस आपने कोई स्टडी किया हो, दैट इज आल्सो यूज्ड फॉर द सैंपल साइज कैलकुलेशन, प्रीवियस रिकॉर्ड, expert opinions, and we will have lot of the software and formula that we will be using in the sample size calculation. तो इसके लिए बहुत सारे सॉफ्टवेयर अवेलेबल है that you can use. The summary of the factor needed for calculation of sample size in social sciences as well. So some of you are from the social sciences, so you would like to do some survey in your community, in your society, in your districts, then how you will go for that one. So same things will apply here as well, as I discussed earlier, कि आपको अपना ऑब्जेक्टिव पता होना चाहिए कि क्या करना चाहते हैं. ओके, उस ऑब्जेक्टिव के अकॉर्डिंग यू शुड नो द टाइप्स ऑफ द आउटकम्स, उससे क्या निकल के आता है, that is the important one. And smallest effects or differences we want to detect, so we want to see that is there any differences, is which is the smallest one. Power, with which we want to detect the effects, and desired level of significance, and the margin of error for estimation and precisions, and expected dropout rates of the subject. Okay, so sometimes we are considering that we will get the results from the 10 subject, but maybe a 2 can be a dropout. So we can add dropout rates along with the your sample size. So उसमें हम add कर सकते हैं sample size calculation में. Now we will talk about the power analysis. So power analysis क्या है? अगर if you can recall the type 1 error and type 2 error, so what is the power? Let's define with the power. So the probability that the statistical test which will reject a false null hypothesis. So basically it's meaning that the probability of detecting an effect given that the effect is really there. ओके तो अगर हम ये जो टाइप 1 टाइप 2 एरर जो आपने किया है, सो टाइप 2 एरर क्या है, we are discussing about the type 2 error. Okay, so the nutshell is bigger the experiment, bigger would be your sample size. Okay, so bigger the power, more likely to be a pick up the differences. So agar power आपका ज़्यादा होगा, it means that you will be considering the more sample size. Okay, so main output of the power analysis is the estimation of appropriate sample size. यानी कि अगर जब हम पावर एनालिसिस की बात करते हैं तो उसमें क्या है, हमारे पास में जो सैंपल साइज है वो अप्रोप्रिएट होना चाहिए, सो दैट पावर यू विल गेट द अप्रोप्रिएट. सो इफ इट इज, अगर आप बहुत ज़्यादा सैंपल साइज लेते हैं तो इट इज़ द वेस्टेज ऑफ द रिसोर्सेज. ओके, सो लेट्स से अगर आपका रिजल्ट जो है आपको 100 सैंपल साइज से ही मिल जाता है, देन व्हाई यू आर डूइंग द 150. Okay, so it's too big, then waste of the resources. Too small, that means you are not able to uh, uh, you are not able to get the answer of the objective. यानी कि आप अपने ऑब्जेक्टिव का आंसर नहीं पा सकते जब आपके पास ज़्यादा सैंपल हो, ज़्यादा कम हो और ज़्यादा कम हो, तो इसका मतलब है कि आप उसको नहीं प्राप्त कर सकते हैं. सो ग्रांट जनरली जब हम प्रोजेक्ट किसी डीएसटी के लिए अप्लाई करते हैं या आईसीएसआर के लिए प्राप्त करते हैं, देन दे विल आस्किंग फॉर द जस्टिफिकेशन ऑफ द सैंपल साइज. द पब्लिकेशन, द रिव्यूअर आस्क फॉर द पब्लिक पावर ऑफ कैलकुलेशन एविडेंसेस. एंड होम ऑफिस, द 3R is required: replacement, reduction, and refinement. So you always think of the replacement means how, what kind of the methods you will bring up, so it can be replaced. So again, you need to think that too small and too big, too large sample size. अगर या तो आप बहुत कम ले रहे हैं या ज़्यादा ले रहे हैं, दोनों ही केसेस में परेशानी है. क्या परेशानी है कि अगर आपने कम लिया तो आपके रिजल्ट्स बेटर नहीं आएंगे, एफिशिएंट नहीं आएंगे. अगर आपने ज़्यादा ले लिया तो आपका रिसोर्सेज जो मनी है और जो वेस्टेज ऑफ ड्रग्स है या जो भी आपका मटेरियल है, दैट इज़ द रिसोर्सेज किल, यू आर किलिंग द रिसोर्सेज. ओके, सो इसके लिए क्या करना होता है हमको, वी विल टॉक अबाउट द अप्रोप्रिएट सैंपल साइज. यानी कि आपका जो भी ऑब्जेक्टिव है उसको अचीव करने के लिए हम कितना अप्रोप्रिएट यानी कि एक एक्यूरेट सैंपल साइज लें, सो दैट इज द यूजफुल फॉर everyone. So there are the lot of the software which is available to calculate the sample size. So some of the free available software, some are the paid software, some are somewhere you can also do in SPSS, in Strata as well. So you need to think of. So these are the software for sample size and power calculations. So Strata is there, SAS is there, NCSS. Okay, so NCSS is the powerful software which is used for the sample size calculations, power and precision. So and query as well, good software for calculating the sample size. These are the software which is freely available. So it's basically freely available means it is an open source software like R. So in R you can also do the sample size estimations. Okay, so you can use G power, you can use PS power and sample size calculations, R package, and optimal design. So these are the sources which is available for calculation of the sample size. Now do you know and do you know what is the meaning of comparison of one sample mean value with the other sample? So do you know the t-test? How many of you are knowing the t-test? Can any one and two participant can tell about the t-test? Anyone can speak? Participants, can you share your idea, what you know about the t-test?
Yes, sir. We have studied T-test and Z-test and ANOVA. This has been completed in the previous lectures or sessions.
Okay, so here I'm just mentioning about the nature of objective or hypothesis. So if it is whatever is your hypothesis, आपने जो भी hypothesis अपने objective के लिए लिया है, it means that it is doing something. So it doing comparison of mean value, comparison of two mean value, paired mean value, or it is correlated, or it is a crossover design, means are many, survival analysis, variance, ROC. So for these all, okay, so whatever the mention here in my slides and more than that, you can have a specific formula for sample size calculations. Okay, so हर एक के लिए जो आपका ऑब्जेक्टिव है, उस ऑब्जेक्टिव से आप किस तरह का स्टैटिस्टिक्स यूज कर रहे हैं, उसके अकॉर्डिंग में you can go for the sample size estimations. Okay, so that is the sample size estimations with respect to your each objective. So each objective is measuring the differences, if correlation, if survival, and then accordingly corresponding formula would be there. Okay, so it is always clear. You should know that this is your objective, and then you are going to do this kind of the analysis. Okay, then similarly, this is another slide which is also telling you the test you are using the comparison of one mean value, comparison of two mean value, or comparison of two mean value with pair samples. Okay, so according to the each things you will be calculating the sample size or power analysis. Okay, so this is the basic things you need to understand for every objective. So if it is somebody is working in the botany, then he and she should have some objective. And what is the objective, statistical objective, that decide that what formula you can use. आपको जो डिसाइड करना है कि कौन सा आपको फॉर्मूला यूज करना है, वो हम डिसाइड करते हैं कि आप क्या स्टैटिस्टिक्स अपने ऑब्जेक्टिव को अचीव करने के लिए यूज कर रहे हैं. तो दैट इज यूज्ड हियर. Now I can go back to the your hypothesis testing or statements. I think it is already covered in your last classes, but still I'm just touching all these things. So there are the two kind of the hypothesis, null and alternative hypothesis. And in the null hypothesis, we are saying that the means of the two treatments are the same or equal. Okay, in alternative hypothesis you are saying that the means are not equal. Okay, Uh, so we will be saying that we are rejecting the null hypothesis or fail to reject the null hypothesis based on the some statistical test and based on the nature of your outcomes. Okay, so while we are making this kind of the decision, so we are doing the hypothesis testing, and then we are committing some kind of the error in hypothesis testing. That is the we are going to study here. So let's say if it is we are testing the new drug is same as the placebo. So suppose my drug is same as the placebo. हम ये टेस्ट करना चाहते हैं कि मेरा ड्रग जो है वो सेम इफेक्टिव है या नहीं प्लेसिबो से. And you are saying that new drug is better than the placebo. This is you want to make the decisions. Okay, so but what is happen statistically? So statistically what you are saying that, so your decision is new drug is same as the placebo, new drug is better than the placebo. That's you are doing. So this is the we are saying that it is the right decision if it is my drug is also same as the placebo, and statistical test is also saying. And if it is you are saying that it is the better than the placebo, but it is the same. So this is basically we are committing error, and here also we are committing the error, and this is the your right decision. So this is the basically the type 1 error and type 2 error. So now we can write, if it is H naught is true and rejecting H naught, that is the type 1 error. And if it is H naught is not true and we are not able to reject, that is means type 2 error. Okay. So type 2 error is nothing but it is denoted by beta, it is denoted by alpha. Okay. So this is, so type 1 error is nothing but null hypothesis is true but statistical test reject it. Type 2 error, the null hypothesis is false but we are statistical test fail to reject. So this we can write about the error in hypothesis testing, that is the alpha is the probability of type 1 error, and this is also known as the significance level. Okay, so this significance level we will be using in sample size calculation. जब sample size calculate करते हैं, then we are using the alpha as well. Beta is the probability of type 2 error, that is means false negative, and power means 1 minus beta, the probability of selecting a good drug. Okay, so 1 minus beta is the power analysis. So here we will talk about that, how you can do the power analysis. So in the hypothesis testing in clinical trials, there are the three types of the hypothesis: superiority, non-inferiority, or equivalence. So I'm not going to in depth in this one. So let's we will talk about the what is the statistical power. Okay, so because we here talking about the samples size estimations and the statistical power. So statistical power analysis depend on the relationship on 6 variables. So it looking the differences of the biological interest, variability in your data means what is the standard deviations, the significance level 5%. Just now we talk about the type 1 error, that means alpha, that means significance level. Okay, then desired power of the experiment. So power means 1 minus beta. Power क्या होता है? 1 minus beta जो अभी हमने देखा था. That is the 80%. And sample size is what number of subject you required. And alternative hypothesis, that is the one sided hypothesis or two sided hypothesis. ठीक है, दो तरह की जो टू साइडेड और वन साइडेड जो आपने हाइपोथेसिस में पढ़ा होगा, that is we are going to be consider here. So statistical power is what here the basically H naught is false but rejecting H naught. So this is nothing but the probability is equals to 1 minus beta. So 1 minus beta is nothing but it is the power of your test. Okay, so this is the correct decision, true positive, that is the your statistical power. Just now we discussed about the type 1 error, that is a false positive. Type 2 error is beta, false negative. Power is nothing but 1 minus beta, and 1 minus alpha is the true negative. Okay, so this is we are going to be discussed. Now the sample size means how much n is required, number of subject is required to do a biological sampling, or if you want to do a survey, so how much sample size is required. Okay, so or how much number of the mice we will consider to know the particular genomic analysis. Okay, so that's where we will talk about the sample size means how much number is required. Power means probability is 1 minus beta. What is the chance of finding an effect that all that really is there? Okay, so basically हम क्या करते हैं? Power means whenever we will have control and the treatment. Okay, so you will be saying that there is a differences between these one. So आपको कितना power चाहिए so that you can prove that your differences. Effect size is basically denoted by delta. इसको हम delta से represent करते हैं. And how big does an effect have to be done in order to find it? Significance level that हमने पहले बताया था alpha, type 1 error, type 2 error, beta, okay, and standard deviations altogether you would be required. So this is also discussed earlier as well. And the statistical power analysis required before the data collections. आप डेटा कलेक्शन के लिए जा रहे हैं, एक्सपेरिमेंट स्टार्ट करने के लिए जा रहे हैं, उससे पहले आपको पता होता है कि हमें क्या पावर एनालिसिस है हमारे एक्सपेरिमेंट का. सो डिटरमाइन दी सैंपल साइज, क्या नंबर है वो निकालना है, नीडेड टू अचीव अ गिवन लेवल ऑफ पावर 1 माइनस बीटा for the designated effect size delta and significance level alpha. Okay, तो ये हम collection से पहले required होता है. Then data collection, but before it analyze, then determine the expected level of power, that is the 1 minus beta, and obtain the sample size n for the designated effect size and the significance level. Determine the minimum detectable effect size, that is means delta we will be finding. Okay, so this is the formula for effect size, standardized and minimum detectable. Effect size क्या होता है, जो आपके पास दो सैंपल्स हैं उनकी मीन वैल्यू क्या है. So what is the differences between the two sample mean values that we are looking. So here we will be finding the mean of one sample minus of mean of other sample, that will give you the effect size. Okay, then standardized effect size, जो differences आया उसको हम अगर standard deviation से divide कर दें तो that is your standardized effect size. And approximately the minimum detectable effect size will be find out from this formula. Okay, so जब हम पावर कैलकुलेट कर रहे हैं तो ये आपको तीन चीजें कॉमनली यूज होती हैं और बेसिकली दीज आर दिस सम हाइपोथेटिकल वैल्यू और बेसिकली कॉमनली यूज्ड वैल्यू. So standardized effect size is nothing but you can take 0.25, 0.5, 0.8. Power generally क्या होता है, 80% power है, 90% power है या 95% power है, that you will decide. Significance level, either commonly we are using the 0.02, 0.05 or 0.01 based on the your research and what types of the your hypothesis that you will decide. How to calculate the sample size, how to decide. So basically I already told you कि differences between the mean value, अगर आप जब mean values को differences देखना चाहते हैं उसके लिए अलग formula. अगर proportion देखना चाहते हैं कि P1 by P2, how many are, let's say there are the 10 smoker out of the 50 student. So what is the proportions? So here we are having the proportion. So proportion के according there is a different formula. Okay, while we are calculating the sample size. जब हम sample size calculate करते हैं, so we do not know the P. That's why we are going to estimate the from the study. So हमें जो value नहीं पता है या तो हम अपना प्रीवियस स्टडी से लेंगे या हम किसी आर्टिकल से इंफॉर्मेशन लेंगे एंड गेट इट फ्रॉम द प्रीवियस स्टडी, डू अ पायलटेड स्टडी. अगर आपके पास में कोई स्टडी का डेटा नहीं है तो आप क्या करिए, पायलट स्टडी करिए एंड देन यू कैन यूज दोज वैल्यू फॉर द फर्दर कैलकुलेशंस. ओके, sometimes we can assume this is to be a 0.5. जो हमें P नहीं पता है उसको हम कंसिडर कर सकते हैं 0.5. If P is 20 to 30%, then choose 30% is the larger sample size. If P is 60 to 8, so this is the some basic criteria which you can consider. Then choose 60%, and if P is lying between 40 to 60, then you can take 50%. So precision means how you can find the precision value. So relative precision assumed to be a 10%, but in case of the rare disease or small p, it may be a more. Okay, so by default we can take 10%, and CI known, then CI by 2, and if it is smaller d is preferred, we have narrowed the CI, and investigator desire to a CI to be a 10%, that means D is your precision is the 0.05, and these are the expected value you can take. So let's say this is the one example where you can estimate the population proportion. So here we are talking about the proportion, यानी कि हम proportion की बात कर रहे हैं कि अगर हमारे पास में objective जो है उसमें हम proportion देखना चाहते हैं किसी भी वेरिएबल के लिए, तो हम उसमें कैसे सैंपल साइज करेंगे. सो दिस इज द n इज z अल्फा बाई 2 एंड पी क्यू डिवाइडेड बाई डी स्क्वायर. सो p आपका 50% है, दैट मीन्स 0.5. d is the margin of error, relative value is 10%, and n is equals to 400. Now you can take some of the examples from the real life problems. So estimating the sample size and power effect size if you are comparing the two samples. So let's say now we can recall the t-test. So t-test is what? If you want to see that the differences between the mean value of two samples. So let's say you will have one sample value is your control, another one is it, another group is the treatment group. तो जब आपके पास में दो सैंपल्स हैं, हम एक एग्जांपल ले रहे हैं यहाँ पर कि आपके पास में दो सैंपल्स हैं एंड यू वांट टू सी द डिफरेंसेस बिटवीन द मीन वैल्यू, उसकी हम मीन वैल्यूज को देखना चाहते हैं, एवरेज वैल्यू को देखना चाहते हैं कि ये सेम है, डिफर है इन कंट्रोल एंड ट्रीटमेंट ग्रुप. ओके, सो दैट्स यू वुड लाइक टू चेक, and that you can find out with the significance label, power, sample size, effect size, and standard deviations. So you know that हम significance label को अल्फा से डिनोट करते हैं, power 1 minus beta, sample size is n, and then delta is effect size, standard deviation you know that it is the s. So sample size you can take the power, effect size. So this is the 0.05. If it is sample size we are finding, then power is 80, and we can calculate it as well. Here the sample size is 128, and it can be 100 power, and 0.5 is your effect size in case of the power, and both you can take this one, and standard deviation is the one that you can take. So let's say how you can do the sample size calculation in case of the statistical package of the social science. So how you can calculate the power or sample size through the SPSS. So I'm giving you a how to calculate the sample size. So let's say you will be going for the analyze, and then you will go for the power analysis. I will be telling you through the software as well. So sample size, then power value is 8.8, and group size ratio is 1, and the mean value of one sample is 0, another sample is the 5, and this is the standard deviation is 1, and significance label is 0.5, and then we click on OK, and this will giving you a sample size. So sample size here is the n1 is the 64, n2 is 64, and power we given here 0.8. So here we already given this is the 0.8, and so this is the calculation from the SPSS. So how to get the sample size estimations. This is an example, and according to the hypothesis for the comparing the mean value of the two samples or the mean value of control and the treatment one. So this is the an example. And now how to calculate the power. So for power के लिए आपके पास क्या क्या होना चाहिए, आपके पास में कितना सैंपल साइज है और कितना डेल्टा है और क्या सिग्निफिकेंस लेवल है, ये तीन चीजें आपको पता होगा, देन यू कैन कैलकुलेट द पावर वैल्यू. So this is the again the same way you can go for analyze, then you can talk about here, you can select the power, and this is the sample one value 64, another b 64, and this is the mean of the group 1 and group 2, वो आपको पता है तो वो आप यहाँ पर डाल सकते हैं और इफेक्ट साइज भी डाल सकते हैं. So this is the standard deviation is 1, and then you can give the significance label, click on OK, and this will give you the power value. Okay, so this way you can find out this power is 0.8, 80% is power for this calculation. So this is done through the SPSS. Now you can do the sample size calculation through the STATA as well. Okay, you can do with the R software as well. R is the open source software. So through that you can also do a sample size estimations. Okay, so this is the how you can write the power to means is 0 and diff is zero point 0.5, and this will give you the n is 128 and per group is 64. ठीक है, तो ये आपके लिए कैलकुलेट कर सकता है. हाउ टू डू द पावर टू मीन्स you can just give and it will give you the power of your analysis. Okay, so this way you can solve the problem of the sample size estimation, but be careful for the sample size estimation. You must need to go back to your hypothesis and what kind of the test you will be using for particular objective, and based on that you will give some value, means from the previous work or previous study, and then after that we can able to calculate the sample size. Okay, so this is the power 2 means is 0, n is 100, and power is equals to n, that you can get the this way you can find out the effect size, means you can decide about what is your delta. Okay, so delta भी आप calculate कर सकते हैं because you know that what is the mean value of one, what is the mean value of others. Okay, so this is the how to this is the presentations which I shown you, but now I will telling you how you can do with the SPSS. Okay, so this is your SPSS software. Okay, so you might be familiar with the SPSS. You can go for analyze and it will be asking the power analysis. आप क्या mean value compare करना चाहते हैं, and then after that you will talk about the independent sample t-test. Okay, तो अगर इसको आप select कर लेंगे and then you can go ahead, it will give you the sample size estimations. So I will telling you again, go on analyze, then select power analysis, then means and independent sample t-test. So this is window is came, then power is there. If you want to calculate the power, so to select power. If you want to calculate the sample size, then you can select the sample size. So let's first we will do a power analysis. So what is the number of n-ones? First group is 64 and another group is 64, and then after that you will talk about the population mean of group 1 is 0 and other is zero point 0.5, and this is the standard deviations, and this is your significance level. Okay, so 0.05 is significance level, and then after that you can go with, you can go click on OK, and now you can see that this is your power value. Okay, so power value is how much? 0.801 is the your power value. Okay, so this way you can find out the your power analysis. Now we will go back and do a sample size estimations. Okay, so this is the means, and then we will go for the independent sample t-test, because we are comparing the two samples mean values. Okay. So you will go on analyze, then power analysis, then mean, independent sample t-test. Okay, so this is the 64 one group, and this is the 64 is the another group, and then after that we will go for the population mean of group 1. And so here first we need to change, now we want to do a sample size. Okay, so power value is zero point 0.8. Here we need to mention group ratio is 1. Then this is the mean value, and this is the your standard deviations, and this is your significance level. Okay, so you need to mention these two. Click on OK. So see here, you will be getting N1 is 64, N2 is the 64, where we use these input values. So these are the your input values. All right, so this you can do with the help of the SPSS. So you can open your SPSS, go on analyze, and then you can play with your whatever the desire is with you. Okay, so this is the one way from you can do a sample size. Okay, now I will be telling you how you can do with stata. So this is your stata. Okay, so here in the, this is the stata, but you need, this is the command window where you need to write the codes. Okay, and you can do a calculation of sample size or power analysis. So suppose, so power is a function. Here power is a function. 2 means difference is 0, and this is the difference is 0.5, and I'm just entering, and it will give you the n is 128, and this is the n per group is 64. So same value I'm considering, which I did in SPSS, and the same value is you can do it in stata as well. So just write this command. So this will giving you a sample size for two samples. Okay, comparing the mean value. So this way you can calculate the sample size. So it is very important what you should know about the sample size. So what you should know means you should know about the previous work which you did or someone already did. Okay, so you can take some references from the previous work, and you can do a sample size calculation. So if you don't have any work, then you can go for the pilot study, which will also telling you how to calculate the and what kind of the value you will be inputting here. So that is the sample size estimations. Okay, so I think this might be helpful for all of you. So now I think, do you want to do a hands-on training or whatever? If you have any other questions, then now it is open for all of you. So this is now uh, next 10 minutes is for discussions. So if you have questions, please ask me. Is that helpful?
Yes, sir, definitely. And I have just one doubt. When we went to the analyze and SPSS, and
Mm-hmm.
independent sample teaches me when you are doing for the sample significance, there was a
Mm-hmm.
ratio you selected one. So is it just when both the pairs are same, then the ratio should be one, or it can be different for different ratios?
So this is the
Yeah, group
we
size
are
ratio. Yeah,
so
yeah.
this is group size ratio is 1. So we are having the same number for the sample 1 and sample 2.
Okay. Means if we have different samples or the numbers are different in both the samples, then we have to change the ratio.
Right.
Okay, thank you.
Any other questions? Even you can ask any research problem if you have any questions, then even you can share here.
So yes, sir, shall we conclude?
Thank you. It was really a detailed, comprehensive, and well presented lecture on power analysis. Now, I would like to invite Shri Utkarsh Khanna ji to present his vote of thanks.
Respected Diwakar sir, and esteemed faculty members and fellow students, it is with immense gratitude that I am here to express our sincere appreciation for the enlightening session you conducted on power analysis. Your expertise and passion for the subject were truly contagious, and you managed to demystify a complex topic in a way that was both engaging and informative. Thank you for taking the time to share your knowledge and insights with us. Your session was not only informative, I would say, but it was also incredibly valuable. We learned the intricacies of power analysis, its importance for the research, and the various techniques involved in conducting it effectively. Uh, I also personally, and I think everyone would appreciate that how you mentioned the significance of sample selection. At the same time, you mentioned the software and also showed the practical that how to conduct exactly the power analysis. So your session was undoubtedly left inspiring and impression on us. We are now better equipped to design a robust and meaningful research studies. Thanks to your guidance, and on behalf of everyone here, I once again express our heartfelt gratitude to you. Dr. Diwakar sir, thank you so much for sharing your expertise and passion with us. We wish you all the very best for your future endeavors. Thank you, sir.
Thank you, Utkarsh, and thank you for all of you.
Thank
So
you.
if you have anything, you can email me anytime. I will try to give you my feedback on that. Okay,
Sure.
so thank you, everyone.
So we will resume with second session at 1:10. Please join by that time. Thank you so much for joining us.